Hello, everyone. My name is Egon Ranghini. I'm a science and technology advisor for 10x Genomics. And today, what I would like to do is give you an overview of our solutions. And so at 10x, we've developed a toolkit that includes three platforms. And this is really key to really understand the complexity of biology. And so we have the Chromium platform that includes our solution for single cell genomics. Then we have the Visium platform that is for spatial transcriptomics. And more recently, we launched the Xenium in situ platform that is for in situ analysis. For today's talk, I'm going to mainly focus on the Chromium platform, mention a little bit Visium, and then in the interest of time, I won't be able to cover much of Xenium. But if you have any question, just feel free to reach out to us, and we can talk more about that. Now, all those three platforms together, they generated 8,200 publication. And that is one of the reasons why we are really the single cell company and the spatial transcriptomic company in this field. And I would say that over the years, we really have developed the most comprehensive single cell portfolio that enables the study of a really wide variety of clinical samples, now including also FFP samples. And so with our single cell solution or using our single cell solution, now we can look at clinical sample at scale. And at the same time, we can significantly reduce the cost per sample by, for instance, multiplexing samples together. And it's something I'm going to talk more about later in my presentation. And so with 10x, essentially now you can interrogate samples with single cell resolution. At the same time, you can also measure multiple features per cells, and this is really critical to understand the full complexity of biology. And now we can also study the spatial context of our samples using two platforms, so our Visium platform and in situ platform, and those provide a single cell scale resolution or even subcellular resolution, respectively. And so going back to single cell to start with, I would say what we have when it comes to single cell is what we call a beginning to end workflow, because everything you need to make a library that can be sequenced on an Illumina flow cell, for instance, those reagents are included in the kit we provide. And so if you want to start a single cell experiment, all you need to do is to prepare a high quality single cell or single nucleus suspension, and then submit it, and then either use your instrument or submit it to your core. Our instruments are present across a lot of different academic cores. Then there is the sequencing, and then there is also the analysis, where we provide fully integrated and fully supported bioinformatics tools, bioinformatics pipeline, and visualization tools for data analysis and data visualization. And importantly, when it comes to software or data visualization, all those tools are free. So you can download them for free from our website and use them for free. Now, when we launched the single cell I'm sorry, wrong button. When we launched the single cell solution, that was 2016, and at that time we could only measure gene expression, which was great, but we knew that because biology is very complex, what we really wanted to do was to develop tools that allows us to measure multiple types of information simultaneously from the same cell. And so over the years, we added application to our single cell kit or single cell chemistry. So now we can, for instance, look at gene expression and simultaneously measure the expression level of cells of the full-length VDJ sequences of T cell receptor or B cell receptor. That is what allows us to identify clonotypes in our sample. We can look at cell surface protein expression, now also actually intracellular proteins. We can measure CRISPR-mediated gene perturbation with single-cell resolution. We can look at region of accessible chromatin, that is to study the epigenetic effect. We can look at isoforms, SNPs, gene fusion. That is by using actually long-read sequencing rather than short-read Illumina sequencing. And just to give you an example of how this technology has been used in the field, I chose this paper, which essentially investigate the transcriptional signature of persisting CD19 CAR T cells in children with BALL, which stands for B cell acute lymphoblastic leukemia, which is the most common childhood cancer. Now, unfortunately, we know that about 60% of children who received CAR T cell therapy experience some sort of disease recurrence. And that's mainly because of poor persistence of CAR T cells in their system post-infusion. And while we know the phenotype of CAR T cells at the moment of production, really little is known about how this phenotype will change over the years after infusion, right? And so to better understand how those change, what those changes are, here in this paper, the authors we used our single-cell immune profiling solution to look at gene expression and T-cell chronotypes of CAR T cells that were isolated from 10 children, and those children were part of a CARPOL clinical trials. And the samples were taken from bone marrow, from the peripheral blood, over multiple time points, actually after 5 years post infusion. And their phenotype was compared to the phenotype of CAR T cell at the moment of production. And what they found, you can see down here in this UMAP, that basically this is 50,000 CAR T cells. We are looking at the gene expression profiling or gene expression profiling data. And we can actually see that the expression profiling changes based on the time point that was analyzed. And we can also see here on the right side, that the most common T cell population across multiple time points were CD8 positive T cells. However, and this was really interesting, it was observed for the very first time, was that at the late time point, which is in green in that the cluster in green in that projection, the late time point was characterized by T cells that were double negative for CD4 and CD8 alpha. And so there was this new double negative phenotypes observed. And this result was actually confirmed by comparing this data to a human PBMC data set that contained more than 200,000 cells. And those cells were also stained with oligotagged antibodies. And so the authors in that paper could simultaneously look at gene expression and expression level of cell surface protein. And so this result shows us that actually the long-term persisting CAR T cell seems to have this double negative phenotype that was not observed earlier or before. Now, in the second part of the study, the authors looked at the T cell chronotype composition. Here, they used our VDJ solution to look at T cell chronotypes. And what the authors found was that most chronotypes across different time points were not observed at other time points. They found that the vast majority of chronotypes common to multiple time points were CD8 positive T cells. However, and this is really important, the vast majority of unique chronotypes, especially those found at the late time points, those were these double negative T cells. And so those results tell us a couple of things. One is, at any time point, pretty much cells were genetically diverse. And also they tell us that the double negative phenotype was not caused by the expansion of, let's say, one or just few chronotypes over time. And so overall, I would say this study really unveils a unique signature for the persisting CAR T cells. Those findings really might help develop more efficient, more effective CAR T cell therapies for the treatment of hematological malignancies. And I would also say that those results were made possible thanks to a single cell approach, or a single cell approach to studying the samples. And if you were to study those samples with a bulk type of approach, let's say bulk RNA sequencing, well, you would have missed most of this information, and certainly this new double negative phenotype. Now, at the next, one of the things we do is we keep innovating. And so recently we launched a new chemistry, which is called GEMX. This is a 3' or 5' chemistry. And what it is essentially is a significantly improved chemistry, significantly improved technology compared to the chemistry we have had so far. And so now, for instance, we can target up to 20,000 cells per sample as opposed to 10,000, so you can get more cells per sample. And the doublet rate has been reduced to half compared to what we had before. And so essentially, you're going to get more single cell data out of your sample. We also wanted to make sure that GEMX was still a multiomic solution, so we can still look at gene expression, cell surface protein, VDJ, clonotyping. We can look at CRISPR-mediated gene perturbation again. We increased the recovery. Now we go up to 80% compared to up to 65 with the previous chemistry. That's really important because you want to retain as many cells as possible once out of your sample. And we also significantly increase the sensitivity. So what you see here is the median gene per cell that you'll detect at a certain sequencing depth. And the new chemistry is the blue line, the old chemistry is the teal line. And so as you can see here, essentially you get a significantly higher library complexity. And so again, more data for your analysis. And then also when it comes to T-cell chronotypes, again, we were able to detect more unique chronotypes. And I have the same data for B cells, but just not going to show here in the interest of time. And last but not least, the new chemistry is also 10% cheaper than the previous chemistry. So really no reason to not to run the new chemistry. Now, just want to go back to this slide I showed you earlier and focus on a solution that we call multiome. And this is what allows us to simultaneously interrogate gene expression and region of open chromatin. So we can study this unique interplay that occurs between gene expression and chromatin accessibility or epigenetic regulation, I should say. Now, with the multiome solution, you start from a single nuclear suspension because we provide chromatin, so we need to isolate nuclei. From this single nuclear suspension, you generate two libraries. One is a single cell gene expression libraries, and then you generate a single cell ATAC libraries. And those libraries can be, or cells in those libraries can be linked together because they will share the same pool of 10x barcode. And so we can use, for instance, the information from the gene expression libraries to annotate cluster in the ATAC libraries, or we can do the vice versa. So we can use ATAC markers to identify gene expression cluster, for instance. And as you might imagine, every single time we add multiple level of information from the same cell, we can also better separate cell population, as I'm going to show you in a second by showing you a paper that use this kit. Before doing that, I also would like to show you that thanks to the multiome kit, we can now look at putative correlation between gene expression patterns and the region of accessible chromatin. What you see here is actually a screenshot of our Lou browser, which is our visualization tool. It's a desktop app, and so it's free to use, first of all, and it's very easy to use. And so here, for instance, you see how region of accessible chromatin in those tumor B cells correlate with the expression level of PAX5. And so there is a really nice paper that I wanted to present, and that uses this kit in the context of trying to improve cell therapy, I should say. And so this is a paper that looks at the differentiation of human induced pluripotent stem cells into insulin-producing beta cells. And as you can imagine, this is really critical for the treatment of type 1 diabetes, for instance. One challenge is for this treatment, or what this strategy, I should say, is that we know that when islets are differentiated in vitro, they don't really have heterogeneous tissues, and so homogeneous tissue. They're more heterogeneous tissue because they present not only beta cell but also alpha cells, delta cells, and also cells of intestinal lineages, such as those EC cells. And so, in order to really make the cell therapy possible, what we really need to do is to better understand how to improve iPSL differentiation into islet. And the key to do that is to look at gene expression and chromatin landscape simultaneously from the same cell. And so, for that, that is the reason why in this paper the authors use the multiome kit to essentially profile iPS derived human islet. And what they found here, as you see here, they found multiple cell populations. So we can see that there are beta cells as expected, alpha cell, delta cell kind of as expected, but we also find EC1 and EC2 cells, and those are cells of intestinal lineage. Now, importantly, here the author stated that EC1 and EC2 cluster, those two subpopulations of EC cell, were only distinguishable, or they could only separate those two populations only when they combined the gene expression with the ATAC information. They were not able to separate out those two populations if they were looking at gene expression only or ATAC only. So that again, that's and highlight again why the reason why you want to try to capture multiple types of information simultaneously from the same cell. Now, down here, we see a region of open chromatins on those three genes and across the different cell population identified. We also see that EC cells and beta cells shares many transcription factors binding motifs. And this was something unexpected. And so the authors wanted to investigate this phenotype a little bit further. To do that, they looked at cell trajectory, and they also reclustered the multiome data set. And what they found was that EC cells and beta cells generated in vitro do not form two separate cell populations. Rather, they form a continuum of cell state that still share multiple gene expression markers and multiple chromatin markers. And that is actually in stark contrast with the profiling of primary human islet, as we see here, where we see individual cell clusters, no EC cells as expected, and also clearly defined region of accessible chromatin. And so there is really a lot of data in this paper. In the interest of time, I can't really cover everything, and so I'm gonna jump on pretty much the final findings and probably also the most relevant findings. And so here, the authors once again use the multiome kit to profile islet that were cultured in vitro for 6 months, iPS-derived islet cultured in vitro for 6 months, and compare the phenotype, compare their multiome profile to islet that were transplanted under the kidney capsule of mice for 6 months. And what they found was that the transplantation actually closed most of or many of the region of accessible chromatins that were observed in the in vitro cultured islet. And that actually led to islet to acquire a phenotype which was more similar to the phenotype of primary beta cell or primary beta cells. And so essentially those results tell us that first of all, the environmental milieu seems to be critical for proper differentiation of iPS cells into islet, and also tell us that the multiome profiling here can really be critical to better understand once again how to improve the beta cell maturation in vitro into islet, and that will ultimately hopefully lead to the design of better cell therapies for the treatment of type 1 diabetes. Now, switching a little bit gear, I'd like to briefly talk to you about a solution we have at 10x that allows us to now fix samples and store those samples for months at -80 degrees. This solution was developed essentially to tackle those 3 challenges you see here. So perhaps limited flexibility when it comes to sample availability, the sample degradation if we fix samples where we stop degradation, and then some logistic constraint. Let's say you are receiving sample from a collaborator. And so this solution is called single cell gene expression flex. With Flex, what you can do is to collect your sample, fix your samples in 4% formaldehyde, and then store your samples at -80 degrees for up to 9 months. That essentially allows you to stabilize your samples, block the biology of your samples, really no matter when you collect it or where you're collecting from. With Flex, we use a probe-based chemistry. So it's not a poly capture. Here we use a set of probes that will recognize all annotated isoform of a particular genes. We look at the whole transcriptome, and so we develop probes to identify all protein coding genes. And because it's a probe-based chemistry, we have a probe panels for human and for mouse. So it's not species agnostic; it's species specific, and that is for human and mouse. Now, we also have a tech node that allows us to either design and spike in custom probes. Let's say you want to identify viral transcript or bacterial transcript or a reporter gene, we can do that. When it comes to sample type compatibility, we can fix almost anything, and we have protocol to do that. And so we can fix cells, we can fix nuclei, we can fix tissue without doing a single cell isolation first. So we can fix fresh or frozen tissue, and we can also isolate single cell from FFP sample. I'm going to talk about that in a moment. Before doing that, I like to talk about that. I mentioned at the very beginning that we can multiplex samples together. So this is what Flex allows us to do. We can combine multiple samples together in the same reaction and essentially profile up to 16 samples in the same lane of a chip and target up to 128,000 cells per sample. So a full chip will be 1,024,000 cells, and those cells are captured in less than 6 minutes. Now, the moment you start multiplexing samples together, that is where the cost per sample significantly goes down. And without going too much into numbers, but you can save up to about 75% running this chemistry compared to doing the same experiment but using the poly capture. And if you want to hear more about that, just reach out to us. With Flex, we can also profile FFP samples. So we can profile single cell from FFP blocks. Same chemistry. What it changes here is how we isolate single cell. So in a nutshell, what we do is we cut a couple of curls or scrolls of the FFP blocks. We dissociate the tissue, and then we isolate single cell, and then we profile single cell with Flex. Now, because we are using or we have access to FFP blocks, we can do single cell. I want to show you really something unique to 10x, and this essentially is the capability of generating both single cell data and spatial transcriptomics data from the very same FFP blocks. And this is by taking pretty much consecutive section of the same FFP blocks. And so now we have this unique capability of generating single cell data and then interrogating the spatial context of the very same sample by generating Visium data using our CytAssist instrument that I'm going to introduce in a second. Before doing that, I'd like to show you this paper, which uses this approach. Essentially, in this paper, the authors generated single cell data and spatial transcriptomics data starting from the same FFP blocks. And this is a colorectal cancer paper. The authors had access to 31 patients with colorectal cancer. They really wanted to create a spatial omics atlas of this cancer type. And so the authors generated single cell data and spatial data from FFP blocks, and they combined the data. And when they combined the single cell and spatial data, they were able to identify a new unique signature that they called immune exclusion signature. This was expressed prevalently mainly in the tumor samples, and some of the genes expressed are DDR1, PAC4, TGF-beta, and so that was used to essentially was actually identified specifically in the tumor cells. Then the authors looked at the spatial transcriptomics data you see here to see where this signature was present across the tissue they were studying. And so As we can see here, the immune exclusion signature was actually colocalizing with this massive region of chromosomal instability, which is, by the way, a hallmark of this type of cancer. The authors also showed that the signature, well, unfortunately, correlates pretty well with poor survival, and so the higher the signature, the lower the survival rate was. We can also see that, for instance, there was a very limited. If we look at the once again at the spatial transcriptomics data, we can see that there was there was very little infiltration of T cell, of CD4 positive T cell and CD8 positive T cells. Now, the reason why I was showing this paper is because, to me, it's extremely exciting to see again an example of using two platforms to study the same sample, and that is really a way to really dig deeper into the biology of your sample, and then it shows you. I wanted to report here the statement from the authors. Now the authors are thinking about using this new signature as a potential biomarker for prognosis and targeted treatment. And keep in mind, this was done by, or the data was generated using FFP sample, using single cell from FFP sample and spatial data from FFP samples. Now, as I mentioned before, we keep innovating at 10x, and now we also have a way to have access to even more samples for spatial transcriptomics, and have a way to streamline the spatial transcriptomics workflow even further. So what I should say actually now is that single cell is a great way to look at sample, but as you may imagine, if you have a tissue, the moment you dissociate the tissue into single cell suspension, well, you lose an important piece of information, which is the spatial context. Now, what we can do with this instrument here, the CytAssist, is essentially, in a nutshell, is to generate Visium libraries starting from tissue section that are they are mounted on regular glass slide. And so in this case, what we can do is to section our tissue, place the section on regular glass slide. The histology part, the H&E staining or the immunofluorescence staining is going to be it happens on those regular glass slide, and so it doesn't require any particular change to a typical histology workflow. Then we use the CytAssist to transfer the analytes from the donor tissue onto the gene expression slide that contains the barcode spot, so that we can capture the analytes. We can barcode those analytes that are essentially probes, very similar to what I showed you earlier for Flex, and then downstream the CytAssist we just need to complete library prep. And with the CytAssist we can access not only to FFP samples, but we can profile fresh frozen or CT embedded samples or fixed frozen samples. Now, thanks to the CytAssist, we were also able to release a new version of Visium, which is called Visium HD. Still a whole transcriptome solution, but now the resolution is up to a single scale level. And that's why and the reason is because we essentially now have a continuous loan of barcoded features, where the size of each feature is only 2 micrometers square. And so if you see here where we have essentially this millions of those barcoded oligos across the capture areas here, the spatial barcode changes every 2 micron in the XY direction, and this is really what gives us this single cell scale resolution of the data. And there is no gaps in between the different spots or different barcoded features. Now, what I'm showing you here is a FFP human colorectal cancer. This was profiled using the first version of Visium, which is now what we call standard definition. Each of those spots is 55 micron in diameter. We can already get a lot of information. We can see different gene expression clusters. We have actually 10 different RNA clusters here. But then if we look at the same sample with Visium HD, now we get way more information because we can go up to a single cell level resolution. And so I'm hoping you can actually appreciate the additional level of information we can gather thanks to Visium HD. Now, I'm gonna finish up here. In the interest of time, just want to say that at 10x really we have this unique toolkit that includes 3 platforms. Those 3 platforms, I didn't have time today to talk about Xenium, but essentially all those 3 platforms are compatible with FFP samples. And so we have this unique toolkit that allows us to profile essentially the same sample by taking consecutive section and thus generating single cell data, spatial transcriptomics data, now again with single cell scale resolution, and in situ data, in that case, will be subcellular resolution. And actually we did that. There are those 2 papers, or they are available online, where we used all 3 platforms to study the same samples. What you see on top of here. Here is a human breast cancer sample, whereas here we study a colorectal cancer sample. With that, I'm gonna finish here. Thanks a lot for your time. Thanks a lot for your attention. And again, if you have any question, please feel free to reach out to us. And thanks, LabRoot, for hosting us. Thank you.